you know what NGS is? NGS stands for Next Generation Sequencing. But why next? Next after what? The reason is that it was developed long after Sanger sequencing, and for a while no newer generation existed. NGS are divided into two major platforms: Illumina, which reads DNA through sequencing by synthesis, and Ion Torrent, which detects bases using semiconductor technology. I'm gonna explain both of them in detail. Let's go step by step. There are 4 stages in Ion Torrent: library preparation, amplification, sequencing, and data analysis. And detection is based on changes in pH. First of all, the DNA is fragmented into small pieces, and special adapters and sometimes barcodes are attached to them. Barcodes are short, special DNA sequences that we attach to different samples when we want to sequence several types of DNA at once. Later, we can tell which sequence comes from which sample using barcodes. After that, the emulsion must be prepared by mixing oil and water. Detergents are added to keep the droplet and prevent them from joining together. Then beads containing complementary adapters, DNA fragments, and aqueous PCR mix are all mixed together while being dispersed in the oil phase. Proper dilution ensures most droplets have only one bead and one DNA molecule, preventing multiple DNAs in the same droplet. I should add that some droplets may contain neither DNA nor a bead, and that's completely fine. It doesn't affect the process. Even though the droplets are inside oil, the whole emulsion is placed in a standard PCR thermocycler, just like a normal PCR tube. In the first step of PCR, the temperature goes up to about 95 degrees Celsius, so the double-stranded DNA separates. Then as the temperature drops for the annealing step, each single strand binds to a primer. One strand attaches to the primer on the bead, and the other binds to a free primer floating in the droplet. Next, during the extension step around 72 degrees Celsius, polymerase synthesizes new strands, one attached to the bead and one free in the droplet. This cycle keeps repeating, and finally each bead becomes covered with many copies of the same DNA fragment. It's important to note that one strand of the original template contained biotin on the A primer at the 5' end, and one of the free primers complementary to the A primer also carries biotin. When emulsion PCR ends, the beads are coated with DNA hybridized to complementary strand containing biotin. After PCR, the emulsion is broken by a special buffer and all beads are released into one solution. This mix contains both empty and templated beads. Then streptavidin-coated magnetic beads are then added to the mixture. They bind to the free strands that have biotin. Since the biotin-streptavidin interaction is one of the strongest non-covalent bonds, these strands are firmly captured. Afterward, the temperature is lowered to allow the two complementary strands to hybridize. One attached to the original bead and the other bound through the biotin-streptavidin linkage. A magnet is used to pull down all the streptavidin-biotin complex and supernatant this empty bead. In this way, beads with the correct DNA template are collected and empty beads are washed away. Then, these enriched beads are loaded onto the Ion Torrent chip, where sequencing by detection occurs. Each well contains a single bead. DNA polymerase, primers, and one type of nucleotide are then added. If the nucleotide matches the template, it is incorporated into the new strand, forming a phosphodiester bond and releasing a hydrogen ion. This causes a small change in pH, which is detected by a sensor under the wall. If the nucleotide is not complementary, no bond forms and no pH change occurs. The Ion Torrent software reads these signals and translates them into the DNA sequence. It's worth mentioning that if there are two identical nucleotides, for example 2 cytosines next to each other, the sequencer shows one peak but higher than usual. Now, let's look at Illumina sequencing. The first step is library preparation, just like in Ion Torrent. The DNA is broken into fragments and adapters are added. These adapters have P5 and P7 sequences that stick to the flow cell. Primers for polymerase and index sequences which let us sequence several samples at the same time are also added. I should mention that flow cell is coated with oligos complementary to P5 and P7. Then the DNA is heated to denature, and single strands are loaded onto the flow cell. They hybridize to complementary adapters immobilized on the flow cell surface. Polymerase, nucleotide, and PCR buffer are then added, and PCR starts to amplify strands. In this stage, bridge amplification happens. Single-stranded DNA bends over and attaches the second adapter on the flow cell. This bridge allows the polymerase to synthesize the complementary strand, creating many copies of the same DNA. At the end of this step, the double-stranded are denatured again, leaving single-stranded DNA fragment attached to the flow cell. Afterward, one of the strands, for example, all of the forward strand, is cut. This cleavage site occurs chemically rather than enzymatically. A specific cleavage site is incorporated at the end of the adapter, and treatment with sodium hydroxide breaks the strand at this site, leaving the complementary strand attached to the flow cell. Now everything is ready for sequencing. When the polymerase, primers, and nucleotide are added, sequencing by synthesis starts. Each nucleotide carries a unique fluorescent label and a chemical block on its 3' OH that prevent further DNA synthesis. As a nucleotide is incorporated into the growing DNA strand, its fluorescent signal is detected by a camera. After imaging, the blocks and dye are removed, allowing the next nucleotide to be incorporated, so the process can continue. If barcodes were added during library preparation, it's necessary to sequence them. Especially index primers are added, and sequencing continues using fluorescent nucleotides to read barcode sequences. What I just described is single-end sequencing, where only one end of each fragment is read. Another technique is paired-end, that both ends of each fragment are sequenced, giving 2 reads per fragment. After the workflow for the forward strand is completed, the same process along with bridge amplification are performed for the reverse strand. Paired-end sequencing is more accurate than single-end, but it's more expensive and is usually used for complicated sequences. Once all reads are generated, the next step is alignment. If you already have a reference genome, the software compares each read to that reference and finds the best match. If there is no reference, another method called de novo assembly is used. Finally, after all reads are aligned or assembled, a clean and accurate version of the DNA is ready for further analysis. Finally, I hope I clarified all of the stages of NGS. If anything's unclear, leave a comment. I read them all and answer your question. If you enjoyed this video, please give it a like and subscribe so you don't miss any of my other videos.